Imagine a world without genetic diseases. A world where inherited conditions like cystic fibrosis, sickle cell anemia, and Huntington's disease are things of the past. This isn't just a dream, it's a possibility that gene editing technology is bringing closer to reality. Picture crops engineered to resist climate change. Imagine fields of wheat that can thrive in drought conditions, or rice that can grow in salty soils. These innovations could revolutionize agriculture, ensuring food security for a growing global population. This is the promise of gene editing, a revolutionary technology that allows us to rewrite the very code of life. By making precise changes to DNA, scientists can correct genetic defects, enhance desirable traits, and even create entirely new organisms. At the forefront of this revolution is CRISPR, a tool of unprecedented precision and power. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, acts like a pair of molecular scissors, cutting DNA at specific locations to allow for targeted modifications. CRISPR has ignited the imaginations of scientists and the public alike, offering both hope and ethical dilemmas. The potential applications are vast, from curing genetic diseases to enhancing human capabilities. But these possibilities also raise profound ethical questions. Join us as we explore the wonders and challenges of this groundbreaking technology. We'll delve into the science behind CRISPR, its current applications, and the future possibilities it holds. We'll also examine the ethical considerations that come with such powerful technology. Like explorers charting a new continent, we stand at the edge of a genetic frontier. This new territory is filled with potential, but also with unknowns. The journey into gene editing is one of discovery, innovation, and careful consideration. We can now manipulate the building blocks of life with astonishing accuracy. This capability opens up new avenues for medical treatments, agricultural advancements, and even environmental conservation efforts. This power to edit genes has the potential to transform medicine, agriculture, and even the very fabric of our society. Imagine a world where genetic diseases are eradicated, where crops are resilient to climate change, and where we can engineer organisms to clean up environmental pollutants. But with great power comes great responsibility. The ability to alter the genetic code of living organisms is not something to be taken lightly. It requires careful consideration of the ethical implications and potential long-term consequences. As we venture into this uncharted territory, we must proceed with caution, guided by both scientific curiosity and ethical awareness. The decisions we make today will shape the future of gene editing and its impact on society. The journey into the world of gene editing is not just for scientists and ethicists, it's a conversation that concerns us all. Public engagement and dialogue are crucial as we navigate the complexities of this technology. Just as we once pondered the implications of splitting the atom, we must now grapple with the power to alter the blueprint of life itself. The choices we make today will have far-reaching consequences for future generations. Understanding CRISPR and its implications is not merely an intellectual exercise, it's about shaping the future we want to create for ourselves and generations to come. Education and awareness are key to making informed decisions about the use of this technology. As we delve deeper into the intricacies of CRISPR, remember this: the future of gene editing is not predetermined. It is shaped by the choices we make, the regulations we put in place, and the ethical frameworks we develop. It's a story that's still being written, and we all have a role to play in shaping its narrative. From scientists and policymakers to educators and the general public, everyone has a stake in the future of gene editing. So, buckle up and prepare to explore the awe-inspiring and thought-provoking world of CRISPR, where the seemingly impossible becomes possible, and the choices we make today will reverberate for generations to come. The journey ahead is filled with promise and potential, but also with challenges that we must face together. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, might sound like a mouthful, but its function is remarkably elegant. This groundbreaking technology has revolutionized the field of genetics, offering unprecedented precision in editing the very building blocks of life. In essence, CRISPR is a molecular scalpel, capable of making precise cuts in DNA, the very essence of life. Imagine being able to edit the genetic code with the same ease as editing a document on your computer. This revolutionary tool is derived from a natural defense mechanism found in bacteria, which use it to fight off invading viruses. Bacteria have evolved this system over millennia, and scientists have harnessed it for genetic engineering. Here's how it works. Imagine a pair of molecular scissors that can be programmed to locate and snip a specific sequence of DNA. This precision is akin to finding a needle in a haystack and then cutting it with pinpoint accuracy. That's CRISPR in a nutshell. But there's more to it than just cutting DNA. The real magic happens in the subsequent steps. The scissors are actually a protein called Cas9, which is guided to its target by a short RNA molecule. This RNA molecule is custom designed to match the DNA sequence we want to edit. This guide RNA acts like a homing beacon, leading Cas9 to the precise location on the DNA strand where a cut needs to be made. The specificity of this process ensures that only the intended DNA sequence is targeted. Once the cut is made, the cell's natural repair mechanisms kick in, attempting to fix the break. This is where the true potential of CRISPR lies. This repair process can be harnessed to either disable a faulty gene or even insert a new gene altogether. By guiding the cell's repair machinery, we can introduce specific changes to the DNA sequence. This ability to precisely target and modify DNA sequences is what makes CRISPR such a game changer in the world of genetics. It opens up possibilities that were once the realm of science fiction. Think of it like this: if our DNA is the software of life, then CRISPR is the editing tool that allows us to rewrite the code. This analogy helps us understand the profound implications of this technology. We can now correct genetic typos, delete harmful sequences, or even insert entirely new lines of code. This level of control was unimaginable just a few decades ago. This unprecedented level of control over our genetic makeup opens up a world of possibilities, from curing inherited diseases to enhancing crop yields. The potential applications are vast and varied. But like any powerful tool, CRISPR must be wielded with caution, as its potential for both good and harm is immense. Ethical considerations and rigorous oversight are essential to ensure that this technology is used responsibly. As we stand on the brink of a new era in genetic engineering, the promise of CRISPR is tempered by the need for careful deliberation. The choices we make today will shape the future of genetic research and its impact on humanity. The journey of CRISPR is just beginning, and its story is one of both hope and caution. As we continue to explore its potential, we must remain vigilant and thoughtful stewards of this powerful tool. The genetic scissors have given us the ability to reshape life itself. The question now is, how will we choose to use this remarkable gift? The potential of CRISPR to revolutionize medicine is nothing short of astounding. Imagine a future where genetic diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease are a thing of the past. This is the promise of CRISPR-based therapies, which hold the key to curing some of humanity's most debilitating ailments. CRISPR offers a level of precision and efficiency that previous gene editing techniques could only dream of. While traditional gene therapy often involved randomly inserting a healthy gene into the genome, hoping it would land in the right spot, CRISPR allows us to target specific genes with pinpoint accuracy. This means we can directly correct the genetic defects that underlie many inherited diseases. The implications for patients with genetic disorders are profound. CRISPR could potentially offer a one-time cure, eliminating the need for lifelong medication or management of symptoms. Imagine the impact on the lives of individuals and families affected by these diseases, freeing them from the physical, emotional, and financial burdens they often carry. Clinical trials are already underway, testing the efficacy and safety of CRISPR-based therapies for a range of diseases, including cancer, blood disorders, and inherited blindness. While it's still early days, the initial results are promising, offering a glimmer of hope for millions worldwide who have long awaited a cure for their genetic conditions. Beyond its medical applications, CRISPR is poised to transform agriculture, offering solutions to some of the most pressing challenges facing global food security. This revolutionary gene editing technology allows for precise modifications to plant DNA, enabling scientists to develop crops that are not only more resilient but also more productive. Imagine crops that are more resistant to pests, diseases, and harsh environmental conditions, capable of thriving even in the face of climate change. These crops could significantly reduce the reliance on chemical pesticides and fertilizers, leading to more sustainable farming practices and healthier ecosystems. This is the potential of CRISPR-edited crops, which could help us feed a growing population sustainably. By increasing crop yields and reducing losses due to pests and diseases, CRISPR technology can play a crucial role in ensuring food security for future generations. CRISPR allows scientists to enhance desirable traits in crops, such as yield, nutritional content, and shelf life, without the need for traditional breeding methods that can take years or even decades. This means that beneficial traits can be introduced into crops much more quickly, accelerating the development of improved varieties. This accelerated breeding process could lead to the development of crops that are better adapted to specific climates and soil conditions, reducing the need for pesticides, herbicides, and excessive water usage. For instance, drought-resistant crops could thrive in arid regions, while salt-tolerant varieties could be cultivated in coastal areas. The benefits of CRISPR-edited crops extend beyond increased productivity. They also offer the potential to improve the livelihoods of farmers by reducing input costs and increasing the profitability of their harvests. By enhancing the nutritional value of staple crops, we can address micronutrient deficiencies that plague billions of people worldwide. This could have a profound impact on public health, particularly in developing countries where access to diverse and nutritious foods is limited. Imagine rice fortified with vitamin A or cassava enriched with iron, providing essential nutrients to those who need them most. These biofortified crops could help combat malnutrition and improve the overall health and well-being of vulnerable populations. However, the use of CRISPR in agriculture is not without its critics. Some worry about the long-term effects of gene editing on the environment and biodiversity. Concerns about potential environmental impacts, such as the unintended spread of modified genes to wild relatives, must be carefully considered. Rigorous testing and monitoring are essential to ensure that CRISPR-edited crops do not negatively impact natural ecosystems. Additionally, ethical questions surrounding the patenting of CRISPR-edited crops and their accessibility to smallholder farmers need to be addressed to ensure equitable access to this transformative technology. It is crucial that the benefits of CRISPR are shared widely and do not exacerbate existing inequalities in the agricultural sector.